Okay, so this is just going to be a quick recap of what we have covered this week on our unit of 3.5 biotechnology. So remember, the first technique that we talked about was PCR, and its main job is to amplify DNA by making many copies from a small amount, and it's going to use a cycle of temperatures to do that, as well as that Taq polymerase that only operates at high temperatures. So we have our dNTPs, our Taq polymerase, our template DNA, and our DNA primers. And the first thing that happens is, remember, we're going to raise the temperature to 95 degrees to separate those strands and break those hydrogen bonds. After that, we cool the mixture down to 50 degrees and allow those primers to anneal. And then after that, once those primers are on there, we can then again raise the temperature to 72 degrees, which is when Taq polymerase takes over and starts making those copies of that DNA. And that every cycle, you're going to get an exponential increase in DNA. So that's PCR. Remember, we also talked about restriction enzymes. So these are enzymes that cut DNA only at specific sites. So each one has a specific sequence that it's looking for. And once it finds it, it's going to cut that DNA, and you're going to get some separation of the fragments after that. Remember, we talked about gel electrophoresis, whose main job is to separate DNA molecules based on size. So we're doing that by applying an electrical field to this agarose gel. And because DNA is negatively charged, it's going to move down towards the positive charge. Remember, the smaller fragments are going to move faster, so they'll come down to the bottom, and the larger ones are going to stay up top. So just a quick word about the restriction enzyme problems that you're seeing in your homeworks. I didn't point this out before, but a lot of times you're going to have a DNA ladder. So you can think of it as like a ruler that's going to tell you how big these fragments are. So when the question is asking you for the length of, or the size of the plasmid, it's referring to these lanes over here that have the restriction enzymes in them, not this one here. So this is just a DNA ladder. So it's telling you this band right here is 2.5. So if there were a band on either one of these that is at this level, it would be 2.5. It's just there as a ruler. It's just reference. So for example, this band here is about 1.5 kilobases, and this one here is about 1 kilobase. So when I'm asking you for the total size of the fragment, you're going to just add these together because this has been cut by Sal 1. So it would be 1.5 plus 1. It's going to give you 2.5 kilobases. So that's how you would give me the plasmid size. So you're going to just take whatever bands you have in one column, and you're going to look over on the side and say, okay, what size does this line up with? And then you're going to add them together. So this one, this matches about 1.5. So this would be about 1.5. And this one is in line roughly with the 1 kilobase. So together, 1.5 plus 1 is 2.5. Now, you might also see lanes with extra thick bands like this. And even though this is at 1 kilobase, there's probably 2 of them in here. That's why it's so dark. So this total here would be about 2 kilobases for this not one digestion. And this fragment here is about 500. So if you were looking at something like this and you wanted the total size, it would be 1 plus 1. So that's 2 kilobases plus 500 is 2.5 total. So that's how you figure out those kind of problems. Remember that DNA profiling is what we call this process when we do PCR on different people to find out if they're the father or if they committed a crime. So we're doing PCR to give us these DNA fragments, and then we're running them on gel electrophoresis in a process called DNA profiling. Remember that we talked about gene transfer. So this is when you're taking a gene from one organism and you're going to move it into another one. So maybe it's a gene that's in humans for insulin because you want to make insulin. Maybe it's the clotting factor for the hemophiliacs. Or maybe it's that GFP jellyfish gene. Whatever the case is, you're going to want to PCR that gene out. However, if you're starting with a eukaryotic gene, before you can move it into bacteria, those eukaryotic genes have exons and introns, and we have to get rid of these because the bacteria does not know how to handle these. So if you're starting with a eukaryotic gene, the first thing that you have to do is make cDNA from mRNA by using a reverse transcriptase to get you that gene. So once you have that, then you can take your plasmid that you get from bacteria, and that plasmid is always going to have some sort of antibiotic resistance gene on it. And you're going to cut that plasmid with a restriction enzyme. to cut it open. And so after that, you can add your gene to your plasmid, mix them all together with another special enzyme called DNA ligase, which is going to join the gene and the plasmid together. So there's two important enzymes in gene transfer, and those are your restriction enzymes, your reverse transcriptase, sorry, there's three, and the DNA ligase. So all of these are needed to do gene transfer. So once you have your plasmid with the gene and your antibiotic resistance, you can then transform it into the bacteria and plate it onto an ampicillin plate. Now, if the bacteria does not have this plasmid, it cannot grow on ampicillin plates because ampicillin is an antibiotic. Antibiotics kill bacteria. So if the bacteria does not have that ampicillin gene, they will die on that plate. So once you have your bacteria that does carry this plasmid, you can then again isolate the DNA from that bacteria and inject it into whatever embryo that you wanted to make transgenic, or you can just produce protein straight from the bacteria. It's your choice. So after that, the gene transfer is complete. Okay, remember that BT corn is a genetically modified plant that contains what is basically a pesticide. So PT toxin is going to kill insects. So it's a pesticide that's in all of the plant tissues. So, and remember the problem with BT corn is that the pollen is going to get on nearby milkweeds, and somehow the monarch larvae are like, you know what, hey, this is poison, I'm going to totally eat this, and then they'll die. So that's the downside of BT corn. Okay, there are also some risks and benefits of GMO crops. I'm not going to read them to you, but you will have to list them if I ask for them. Okay, remember there's different forms of cloning. Some of them are natural, like Hydra, which can be cloned by budding, that you see here. And then there's also the planaria that can be cloned by cutting, or the aphids that can reproduce offspring via those oocytes that are generated from mitosis in a process called parthenogenesis. So if I ask you to list them, you can't just say Hydra, planaria, and aphids. You actually have to list, like, Hydra can be cloned by budding, planaria can be cloned by cutting, aphids can be produced, offspring via oocytes generated from mitosis. It has to be the whole thing. So same with the natural cloning in plants. You have to be able to tell me that replanting a garlic bulb is going to get you a new identical plant, and explain that the strawberries grow those long stems called runners, which produce genetically identical plantlets. And you'll have to give me the list of stem cutting, and then say you cut the samples at the stem and then replant them in a growth medium, so where it grows new roots and gives you a genetically identical plant. So remember that there are artificial forms of cloning, one of which is embryonic splitting. So that's when the embryos are physically cut. They do this in cows to make genetic clones of cows. And the most important one is the somatic cell transfer. So remember, you start with donor nuclei from a differentiated adult. So this is different than what we have over here. So for embryonic cloning, you're splitting the actual embryo, and that's the clone. For somatic cell nuclear transfer, you're cloning an adult that's already walking around in the world using differentiated tissue. So you take that nuclei from the donor, you then go get eggs from some sort of surrogate somewhere, and you're going to treat those embryos with UV irradiation and remove the nuclei. So that's all to get the nuclei out of there, because you don't want to have any DNA from the donor, you just want the clone. So once you have your eggs prepared, you can then insert your donor nuclei that you got from your cat or your sheep or whatever, and inject those into the egg. And then at that point, to get these to start growing, you apply an electric shock and transfer all of those cloned embryos into a surrogate. So then these babies would be genetically identical to what you started with over here. Okay, so that's the recap. Good luck on the test tomorrow. You guys got this.